Thank you very much to the organizer for this wonderful event. I'm doing PhD on interhost differences in immune response against Histomonas meleagridis. This is the first work package of my PhD. So to proceed further on my PhD, I need the in vitro culture of Histomonas meleagridis with enhanced growth. So Histomonas meleagridis is a unicellular anaerobic protozoan which often affect the kika and liver of the poultry birds, and it is mainly present in the kika as a flagellated form, while in the liver it is present as amoeboid form. As you can see in the figure 1, the kika lumen form, and in the figure 2, the tissue dwelling form. Histomoniasis is an intestinal protozoal disease of gallinaceous birds. It is also known as blackhead disease, infectious enterohepatitis. It is occur when histomonads penetrate the caecal wall and then enter the bloodstream, then parasitize to the liver of the bird. This disease first documented in 1893 in Rhode Island of USA, then in Japan in 1905, and by 1926 in EU, Asia, and other Australian countries. To date, has been reported worldwide. As far as we discuss the burden of this disease, it is mostly affect to the turkey birds where the mortality rate is 100%, and death occurs after 2 weeks of infection. In chickens, it affects milder on health, but it decreases the egg production and growth rate of the chickens. So before 2000, we were using the nitroimidazole and nitrofurans to treat this disease, but due to the potential carcinogenic effect and concern about residues in food products, many countries banned these drugs. So until now, we don't have any approved drugs in the market. That's why this disease reemerged with up to 100% mortality in turkeys. As you can see, the study overview on Histomonas is increased from 2000 until now. The objective of my research was to isolate the bacterial culture from caecal content to compare the growth kinetics of Histomonas meleagridis, and then to establish in vitro culture for enhanced growth of Histomonas meleagridis. In the methodology, we first isolate the bacteria from caecal content. We get the turkey's caecal content from commercial farm and then inoculate into the nutrient broth and then loop streaking on agar. Then we identify the single colony by subculturing through DNA extraction and then performing PCR with primer V1, V9, then purify these PCR products and sequencing by using 16S RNA sequencing. For the in vitro culture, we use the anaerobic chamber. We made diets media and use rice starch, inactivated horse serum, and incubate into the anaerobic chamber for 24 hours. Then we mix Histomonas meleagridis and incubate for 2 to 3 days at 40 degrees centigrade. And then we identify in macroscopic examination and count these cells by using Newberg chamber. We also identify the Histomonas by using PCR and sequencing. For the results, we got good results by using the isolated bacterial culture. When we use the whole bacterial culture isolated from turkey caecal contents, we got a sustainable in vitro culture of Histomonas meleagridis. As you can see in the graph, in different passages, we got a sustainable growth of Histomonas meleagridis, which is enough to infect to the further study. When we use the E. coli as a bacterial source for the culture of Histomonas meleagridis, we got a good results, but as compared to whole bacterial culture, we got less quantity of our Histomonas meleagridis. When we use Staphylococcus epidermidis and Staphylococcus hominis, we got a few results, but when we go further for the many different passages, we we can't get the enough concentration of Histomonas meleagridis for our further infection. And same for the Salmonella enterica and Enterococcus faecalis. We also try to get more growth of Histomonas meleagridis by using methyl cellulose and different starch mixtures. As you can see in the figure, when we use the methyl cellulose, we got a good results as compared where we can't use the methyl cellulose. In the different starch mixtures, we use rice starch, potato starch, corn starch, and rice and potato starch in combination. We got the good results when we use the rice starch as compared to other different starch mixtures. So the key findings of this research work package, we got high yield of in vitro culture of Histomonas meleagridis by using whole culture bacteria isolated by fecal content. Use of methyl cellulose in diets media increased the growth of Histomonas meleagridis. Rice starch produced high in vitro culture Histomonas meleagridis as compared to other starch mixtures. So my future roadmap is to test our in vitro culture produced Histomonas meleagridis by developing in vivo model. So we will try to get the best effective route of Histomonas infection. As you can see in the in vivo model, we will grow the turkey birds and after on 70 days, we infect them with our in vitro culture produced Histomonas meleagridis. Then we slaughter to see the kika and liver of these infected birds, and then we will proceed further for our main project. So you are welcome to the Animal and Human Health Engineering Host Pathogen Interaction Department of Biosystem, Faculty of Bioscience Engineering, KU Leuven. My promoter, Professor Jan Pasches, he is working on different parasitic, viral, and bacterial diseases. My assessor, Professor Flip, he is immunoparasitologist from Riga Institute, KU Leuven, and Professor Sam, he is working on microbial and plant genetics. Thank you very much.